So this is demonstrating the staining for oxidized LDL in the top and for myeloperoxidase in the bottom. Okay, so the notion would be then that MPO is a critical role, has a critical role in modifying LDL to a form that can bind to scavenger receptors and promote foam cell formation. And all of these events have been mimicked in the test tube using LDL and myeloperoxidase in macrophages. So when I moved from Washington University to the University of Washington, we became interested in the idea that HDL, or the good form of cholesterol, might also be targeted for oxidation. And this is surprisingly something that no one had ever really considered before. Almost all the previous work on oxidative stress and damage of lipoproteins had centered on LDL. So one of the ways that we investigated this question was to measure a very specific product of bleach. So one of the great things about studying bleach is that it's a very unusual oxidant. It contains a reactive moiety called chlorine. Chlorine can react with molecules to form chlorinated biomolecules. And so what we did was to develop assays, very specific and sensitive mass spectrometric assays that could detect chlorotyrosine in human biological material. And then we went on to use this technique to look at HDL isolated from humans. So the notion would be this chlorinated tyrosine is a very specific molecular fingerprint that helps us identify whether or not this chemistry is taking place in the human artery wall. So this is just illustrating the chemistry again. Myeloperoxidase reacts with peroxide to form bleach, and bleach chlorinates an amino acid called tyrosine to form 3-chlorotyrosine. And then, in turn, we suspected that the bleach might react with the major HDL protein, which is called ApoA, to form chlorinated tyrosine residues. So the initial experiment was actually a very simple one. We isolated HDL from the blood of either people with established coronary artery disease, abbreviated CAD, or age and sex matched control subjects, and then we quantified the levels of 3-chlorotyrosine in the HDL using isotope dilution gas chromatography mass spectrometry. And we really got a rather striking result. When we looked at the patients with established heart disease, every single subject had detectable chlorotyrosine in their HDL. In striking contrast, except for one individual, chlorotyrosine was undetectable in the HDL of the subjects who were disease-free. So based on these and other studies, we proposed that HDL was also a target for oxidative damage in humans with coronary artery disease. So one of the ideas for what HDL is doing is, as I said earlier, is that it's removing cholesterol from macrophages. And so it was an attractive hypothesis to us that this kind of chlorinated damage might block the ability of HDL to remove cholesterol from cells. And so we went on to investigate this question with our colleague Jack Oram, who provided those highly detailed molecular models of good and bad cholesterol that I showed you earlier. And we focused on a specific protein in macrophages called ABCA1. So this is a simplified view of how HDL removes cholesterol from macrophages. So the pathway starts with the release of lipid-free ApoA1, the major HDL protein from HDL. So most ApoA1 in the circulation is associated with HDL, this lipid protein complex. But it turns out that that complex will not remove cholesterol from cells by this pathway. Instead, the ApoA1 has to come off of the HDL particle. In turn, this, what we call lipid-free ApoA1, interacts with a protein that's in the membrane of the cells to promote the efflux of cholesterol and other lipids in the membranes from the cells. And so what happens is these cells actually generate little lipoprotein-like particles at their cell surface that are taking away the cholesterol together with other lipids from the cell surface. And this in turn depletes the macrophages of cholesterol. We know this pathway is important in humans because when you lack this ABC protein, you suffer from Tangier's disease. And in this disorder, you accumulate cholesterol-laden macrophages in many different tissues in your body. And we can also mimic this disease in mice by knocking out the ABCA1 pathway. And when we do this just in macrophages, we dramatically increase atherosclerosis. So this is illustrating in a little bit more detail how we think this is working. So as I emphasized earlier, it's a lipid-free form of ApoA1 that has to interact with ABCA1. And this is actually a representation of a more detailed model of that structure based on X-ray crystallography. And the key feature here for you to observe is that it is composed of a 4-helical bundle. So if you look carefully at the left side of the figure, there are 4 bundles that run next to each other in an anti-parallel conformation. This is a key structural component of the lipid-free HDL. In contrast, when ApoA1 is bound to HDL, it has a very different conformation. Now it looks like a horseshoe-shaped molecule, and one could just envision this molecule going around a spherical particle and accompanying it. So what AGs et al. Proposed in a paper that was published several years ago was that the key step in lipid-free ApoA1 removing cholesterol from cells was a reorganization of the lipid-free ApoA1 from this 4-helical bundle structure to this horseshoe-shaped structure. And they proposed that remodeling of the particles in the hinge domains was critical for this pathway. So what we were able to demonstrate in a whole series of studies that I won't have time to show you tonight is that specific amino acid residues in these hinge domains of ApoA1 were specifically targeted for oxidative damage by myeloperoxidase, and that this blocked the ability of the chlorinated ApoA1 to remove cholesterol from the cells. And so the idea is that by targeting very, very specific amino acid residues in this protein, we can disrupt its biological function. Okay, so just to summarize the story so far, what we would argue then is that myeloperoxidase, which is actually secreted by the macrophages themselves, can oxidize LDL, and this causes it to bind to the scavenger receptors and deliver cholesterol. And in parallel, it blocks the ability of HDL to remove cholesterol from the cells. And this kind of double whammy plays a key role in promoting macrophage foam cell formation in the artery wall. Okay, so the traditional notion then